My name is Vladimir Bashkirov, and I will present you the idea of the patent for the invention of the novel method for single-molecule DNA sequencing and the device for its implementation. It is known that during DNA synthesis reaction, the enzyme DNA polymerase inserts a nucleotide complementary to the nucleotide in the DNA template, which is accompanied by the release of pyrophosphate and the hydrogen ion, red arrow, into the solution. In this case, the screening time for the hydrogen ions by solution of counter ions is several microseconds. The essence of the invention is based on the ability of single electron sensor to respond with a current pulse to the movement of one hydrogen ion in solution, which is released each time a nucleotide is inserted by DNA polymerase immobilized on the surface of the sensors. Thus, the sensor registers the facts of nucleotide insertions. To find out the name of each of four types of the nucleotide, A, G, T, or C, inserted into the growing DNA chain, we propose an algorithm for restoring their names, which is shown in this figure. The statistically average duration of the time intervals before the incorporation of a nucleotide is inversely proportional to the concentration of type of the nucleotide that is being inserted. If the concentration of each type of the nucleotides in the reaction mixture has a predetermined value, for example, concentration of A is higher than C, higher than T, and higher than G, then the delay times before insertions of the nucleotides will be described by the inequality shown in the figure: time for G more than time for T, more than for C, and more than for A. During DNA sequencing, the nucleotide sequence assembly electronic circuit calculates all time intervals between nucleotide insertion signals and compares them statistically both with each other and with the distribution parameters of the diffuse movements of nucleotides in solution, which are set for each type of nucleotides before sequencing. Such comparison by the electronic circuit results in generation of nucleotide sequence of DNA fragment, as shown in the bottom of this figure. The next figure shows a diagram of the DNA sequencer, the instrument that implements the method of single-molecule DNA sequencing of present invention. The main module of the DNA sequencer is a CMOS microchip with an array of sensor cells which generates raw data on the nucleotide sequence of each sequenced DNA fragment. All other modules provide a supply of solution, control the chip performance, and process raw data. The DNA sequencer can be designed as a portable and autonomous device which can be used at the point of need and is capable of obtaining sequence of human genome with an accuracy of 99.999% in about 4 hours at the cost of $100.